University of Glasgow. And I'm going to be joined this afternoon, chairing this afternoon's session by Valerie Cwik from the Muscular Dystrophy Association, who's in the program. We're also going to be joined by John Porter from the NIH, who unfortunately has not been listed in the program, but he has been listed in the contacts list as coming from Cape Canaveral, which sounds much more exciting than coming from from the NIH. But he'll be joining us and doing the session this afternoon with the advocacy groups. So we're going to start off in this interactive session by having some reports from the meeting that's been taking place over the last few days from the scientific part of the meeting. And we've discussed a lot of things over the last few days, lots of exciting developments in terms of the basic research that's going on, understanding more about the clinical aspects of the disease, and how these things can come together to develop new treatments that we hope will be coming online within the next few years. So there's been a huge amount of data presented, and the three speakers in the first session this afternoon have a tremendous challenge to try and condense all of that information and get over to you some of the exciting developments that have happened. So our first speaker is going to be Mani Mahadevan from the University of Virginia, and he is going to try and summarize disease mechanisms in myotonic dystrophy. Mani.
Good afternoon, everybody. I want to thank the organizers for organizing this meeting. It's been a terrific meeting. We've heard lots of new and exciting things in research, in basic science, in clinical science, as well as some therapeutic development, and you're going to hear a summary of all of that. I want to personally thank T and Maury for organizing a wonderful meeting, and I will get started. So, where do I get the slides, guys? Yeah. Are we working here? All right. Okay, so I want to start off with a little historical perspective. This month, for those of us old-timers who have been working on myotonic dystrophy for a while, happens to be the time 20 years ago when the labs that were working on discovering the gene were actually finding the gene and the mutation. I mean, I'm talking about these days, right now. It was really exciting. And so I want to take you back a little bit. I talked to Jeremy Kelly about this. I said, why don't you walk us back a little bit and kind of go over where things were and where we are now. So back in 1991, well, I'll tell you who that is later. So, and he knows. So back in 91, the Human Genome Project was just underway. You know, it started in the middle of 1990. The idea was that we're going to sequence the whole genome, and myotonic dystrophy was actually one of the projects of the Human Genome Project. You can see what cell phones looked like back then, and also what a laptop computer was. And there's somebody who looks rather familiar down there. So Weird Al Yankovic, right? So this is the last sentence of the paper that we wrote on the discovery of the DM mutation in Science. So in light of the surprising result that the DM mutation is found in the 3' non-coding region, that means the 3' UTR of the DMPK gene, it will be interesting to examine the role of this defect in the pathogenesis of myotonic dystrophy. Talk about an understatement. I think we were hedging our bets. So it took about 10 years to go through a whole lot of experiments and a lot of concepts, because that was a very different mutation, kind of broke the rules of genetics and opened a whole new area of biology. And it took a long time, but it took about 10 years, but we figured out by that point, by 2001, we had discovered things like RNA foci in DM, the CUG binding protein, Dr. Swanson discovered the muscle-blind 1 protein involvement in DM. And RNA splicing defects were reported by Dr. Cooper's lab back in 1998. And the first mouse model of RNA toxicity came out of Dr. Thornton's lab. And then in the middle of the summer of 2001, we heard about the DM2 gene, and that really kind of solidified the idea of RNA toxicity. Okay? And by that point, the human genome had been sequenced, and it cost $2 billion. And now you see what cell phones were looking like in 2001, and what laptops look like. Okay, let's move forward another 10 years. Some things don't change. Okay, so now we have a lot more mouse models. The concept of RNA toxicity is well established, and we've now even shown that RNA toxicity is reversible using mouse models. We have treatment development with regard to antisense oligos, morpholinos, small molecule drugs like pentamidine, which you've heard about, even gene therapy has been tried, and now you can sequence a human genome for $5,000. And now you see what cell phones look like, and you can hold a laptop with just your thumb. And Weird Al hasn't changed, but you know, I was thinking about our visit to the museum yesterday, and the appearances can be deceptive. Look at that. Isn't that striking? Okay, so I'm going to highlight some of the things that came out of the basic research in the first couple of days. RNA toxicity, so I have about 8 things that I want to mention quickly. So RNA toxicity is getting more complicated. We have more mechanisms to consider. There are more RNA binding proteins that are involved. There are more functions for the MBNL1 protein, more mouse models. And then there are issues with the repeat expansion and what's happening in blood versus what's happening in tissues. And there was some interesting discussion about something called a progenitor allele, which is a way of measuring the repeat size that one actually inherited early on, and what its value might be in diagnostics. So I'm just going to mention that. And there are also more targets that have come from all this research for potential therapeutic intervention. This building is the Burj Khalifa in Dubai, so let me talk a little bit about RNA toxicity getting more complicated. So one of the things that we heard about at this meeting is that we've all heard about the CUG RNAs and how they get stuck in RNA foci. But now there's an idea that you might actually have RNAs coming off the other side, other strand. We may have CAG RNAs in patients as well. And that's been complicated even a little bit more by something called RAN translation. So the idea is that there are these toxic proteins that are being made also in not only myotonic dystrophy, but perhaps in other triplet repeat disorders like Fragile X we heard about. MicroRNAs, they're a new kind of RNA that has exploded in biology over the last decade, and now we're finding out that microRNAs may be affected in myotonic dystrophy. Now microRNAs are RNAs that are thought to be involved in fine-tuning gene regulation and perhaps involved in stress response. And there was an interesting presentation at this meeting suggesting that MBNL1 sequestration in RNA foci might actually affect microRNA processing in DM tissues. And of course, people have reported a lot more genes that are being expressed aberrantly in myotonic dystrophy as well. Now, there's some new RNA binding proteins that have been shown in DM. So one of them called SMAUG, SMOG, has been shown to potentially suppress RNA toxicity by interacting with CUGBP1. Several other proteins have been mentioned, including stofin-1, another protein called P68 RNA helicase that was mentioned by 2 groups, and FOX1 and 2. So these are all proteins that might modulate RNA toxicity. So they present opportunities for understanding the disease better, as well as potentially for therapeutic intervention. More functions for muscle-blind. Well, I just told you about muscle-blind playing a role in microRNA processing, and there was a study presented that suggested that this could be playing a role in the cardiac manifestations of DM. And then there was another very interesting several presentations that talked about other roles for muscle-blind. So we all hear about muscle-blind and its role in RNA splicing and how it's bound to the RNA in foci, but there's been some presentations that suggest that it might play a role in moving the RNA within the cell from one place to another, and that would then be important in where proteins are made, when proteins are made, and how much is made within cells. There were more mouse models of muscle-blind knockouts. So we had papers presented where muscle-blind 1 and 2 were knocked out, and muscle-blind 1 and 3 were knocked out. And we saw with those studies that that removal of more muscle-blind caused a more severe phenotype in the mice. And some of these phenotypes may be related to some of the things that we see in myotonic dystrophy, that like some aspects of congenital DM, or perhaps some aspects of the neurologic manifestations of DM. Also, a very interesting presentation from Dr. Ranam's group showed that you can make a mouse that makes a lot of muscle-blind, and in fact, the one thing that they noticed was that as soon as the mice are born, they seem to have like an adult muscle right from the get-go. But otherwise, the mice live for a long time without any adverse effects. So as you know, there are some therapeutic strategies where people are suggesting that we increase the amount of muscle-blind. There was some concern that increasing muscle-blind might have adverse effects, but this study seems to suggest that at least in these mouse models, that was not the case. So that's promising. There were many more splicing defects identified and reported, but as you know, we now have a long list of splicing defects, but what's even more important is to try to identify and understand if these splicing defects really have a role to play and what their specific role is in myotonic dystrophy. We have good evidence for chloride channel and its role in myotonia, and we have strong suggestive evidence for its role in the insulin receptor. But we now have evidence that some of these splicing abnormalities, based on studies done by other groups published elsewhere, that these might be playing a role in muscle weakness, and there's a report of a channel that's misspliced in the heart that might be playing a role in conduction abnormalities. Repeat issues and tissues. So muscle, we heard from several groups that muscle, brain, and I think even heart have much higher repeats than what is diagnosed or seen in the blood cells. So I run a DNA diagnostics lab, and people get upset at me because I say, I'll tell you if you're expanded or if you don't have an expanded repeat, but I'm not really a big fan of telling people exactly how many repeats they have. And what these studies showed was that what's in the blood is not necessarily what's happening in the tissues that are affected in the disease, okay? So someone might have 100 repeats in their blood, but they may have 1,000 repeats in their muscle, or 1,000 repeats in their heart or brain. So, and that might be the thing that's more relevant. So I just want to put that out for folks to think about. Then, as I mentioned, the other thing was this idea of the progenitor allele. And one of the first talks we heard at this meeting was from Darren Monkton's group and Dr. Botas, is that right? Morales. Morales' group in Costa Rica, who did some studies on progenitor allele sizing and suggested that this might be predictive of risk for congenital DM. Now, as I said, I run a DNA diagnostic lab. My ears perked up when I heard that. So I want to learn how they do this and see if there might be a potential clinical application for something like that. We also heard from people who work on DM2 that they've identified families where the repeat sizes are almost in the normal size range. And so that is puzzling, but it needs to be investigated some more. But as you see, many different things are happening in the field. People might recognize this picture. If any of you saw Lord of the Rings, these are the remarkable mountains in Queenstown, New Zealand. The research has also led to development of biomarkers and potential targets for therapy. So some work, in fact, the first thing I'm mentioning is things that came out of our lab. We identified a receptor that's expressed in the skeletal muscle in both mouse models and in patients that might be a target for a biologic therapy. I think Dr. Berglund might talk about that some more. We also heard this morning about MBNL1 decoys. That is shortened versions of MBNL1 that could be used to potentially displace muscleblind. So the biology of that needs to be understood better as well, because we need to know if these decoys might have deleterious effects on other things that MBNL normally does. And then, of course, there are a lot of new cell lines and animal models that have been developed. And I want to take this opportunity to emphasize to those who fund this research that the importance of allowing this kind of work to go on in things like C. elegans or fruit flies or zebrafish, because those model systems have been used for drug screens and have been successful in identifying some potential compounds. The other thing, IPS that I mentioned, are stem cells that have been derived from patient cells that could be used in testing and developing drugs. Okay. So the ship's been sailing now for 20 years, and but before we had no idea where we're going. But now we have a good sense of where we want to be, and you can see the rainbow in the background there. So maybe we're closer to the pot of gold at the end of that rainbow. And I want to emphasize that in order for the research here to be successful, I think the first slide that was up there said that we need to continue the partnership between those of us who do research and those of you who are here to hear about what we're doing. And I really appreciate the fact that you're all here. And it is important for those of us who do this research to interact with the patients who I think we're serving. I think that's really what this is all about. So 20 years ago, when we were cloning this gene, the reason that cloning this gene was successful was because we developed that relationship with families and talked with them, met with them, got their DNAs, diagnosed them when there was no DNA testing, and that allowed the gene to be cloned. It could not have been done without the families. And the same thing holds true for the development of therapy. So we need you guys to partner, and we need to talk, and we need to communicate with each other and work on this together. So that's what I have to say. Thank you.
Thanks, Dr. Mahadevan. I think we're going to hold questions until the Q&A session a little bit later this afternoon. And before I introduce our next speaker, I'd like to mention that this afternoon's session is being live streamed, so we welcome all the families who are joining us around the country and around the world. So our next speaker, who's going to give us an overview of the clinical science highlights from the presentations at this meeting, is Dr. T. Ashizawa, who is at the University of Florida in Gainesville. Dr. Ashizawa was instrumental in making the IDMC meeting happen along with Dr. Swanson. So we thank him for that and welcome him to give us the highlights.
Well, thank you, Valerie. Wow, so many people. That's great. And, well, let's see. Can we get the first slide? All right. So this is a beach in front of us. This is from hotel website. And, you know, when I proposed the venue of Clearwater about 2 years ago, in Würzburg, that was the last meeting of the IDMC-7, I promised white sand and blue sky. And I was really worrying about white sand about a year ago with BP oil spill. We may have black sand. And you know, earlier this week there was a little cold front coming and a sort of a cloudy overcast. So I was really worrying, but thank you to all of you. We have blue sky now, so please enjoy for the next few days. Okay, so I'm going to talk about the clinical aspect of myotonic dystrophy, what kind of advances have been made. Dr. Peter Harper, Professor Harper, I don't know where he is in this audience, that right there, over there. Professor Harper has done a lot of clinical work, and he published books and legendary books of myotonic dystrophy by Peter Harper. And I thought that everything has been described by him, but wrong. There are still things are coming out, and there are a number of new findings, but particularly in the area of juvenile onset of myotonic dystrophy, and DM1 and DM2 distinction, and other things. Now, so I'm going to just go over several different areas, and but particularly first the clinical characterization. So this is a slide from Dr. Benedict Schoser, who was giving the overview before the clinical science session took place during the scientist session. And that really the problem is that still the mortality is an issue. Lifespan is truncated in both DM1 and DM2 to some extent, but particularly DM1. And that when you see that congenital myotonic dystrophy or childhood myotonic dystrophy, it's even exaggerated in the lifespan problem. So DM2 is still, there are some sudden deaths and mortality involved, but in general, life expectancy is pretty good. Now in congenital myotonic dystrophy, this is substantially rare, but this is really hallmark of childhood myotonic dystrophy. It's a neonatal onset or even in utero. While the child is in the womb of the mother, it starts with a low level of fetus mobility. And they tend to be born a little bit earlier with hospital stay average of 49 days. And they have relatively high mortality, but during that period, oftentimes doctors and family decide to terminate the ventilation because it looks so grim. You can't really imagine those babies can survive. But surprisingly, when they survive, they do extraordinarily well. They start showing the progress in both motor and intellectual milestones, although there's substantial delay, but they clearly advances in those areas. So that we had some question whether making a decision at 1 month after birth, is that really premature or it is a good thing? And I think many of you know patients with a congenital form of myotonic dystrophy, they are lovely people. So, it's a very hard philosophical question, but still it's a lingering question. Now, DM2 has very similar parallel findings with DM1, with in general relatively mild symptoms with no congenital myotonic dystrophy. But they're having cardiac death and etc. In this meeting, already Dr. Mahadevan talked about, but they're having a couple of families with very short CCTG repeats. So that gives a sort of diagnostic dilemma. And sometimes, in the cases from Finland, the repeat is less than 70 repeats, 70 CCTGs. And in German cases, also very short. But in German cases, if you look at very carefully with the special kinds of assays beyond the regular PCR diagnostic testing assay, you can see the expanded repeats, very large ones as well. So this poses some new diagnostic problems with DM2. Now I'm going to just go over the systems. I'm not going to talk about some of the very important organ systems. There was a poster by Rochester Group about GI. GI is probably the most commonly addressed complaints by the patients, and along with muscle weakness and myotonia. But in terms of the concern, level of the concern, GI symptoms are very important. But from the viewpoint of socio-economical impact, for example, the employment, social security, disability, and actual functionality, brain is an important part of the disability. So myotonic dystrophy is a myotonic muscular dystrophy. There is no question about that. Muscle is a very important part of the disease, but brain is also a hidden, very important problems. So as I said, it's not the top 5 concerns of DM patients, but it's a very important concern of myotonic dystrophy patients, and there are a whole host of manifestations listed there. Those abnormalities are not, it used to be said that brain problems may be just very elusive and not really, you cannot really pinpoint, but at least in adult myotonic dystrophy patients, very specific functions and areas of brain seem to be affected, and those are being characterized by different techniques, neuropsychological testing, and that can really specify that defect, as well as functional MRI studies and other imaging studies. The big question is, congenital myotonic dystrophy patients, they have mental retardation, but as I said, they can show the progress. So they do not, at least in the beginning, they don't have any degenerative or progressive brain disorder. So they actually get better in their function. But in adult, there have been debate whether this is a static problem or it is a progressive problem due to the neurodegeneration. So that is not really completely settled. There are 3 major approaches you can take. One is a structural measurement. That's by MRI or autopsy brain. And if you look at those, you can count the volume of the brain. In general, the volume is somewhat smaller, and there are white matter changes, and ventricles, which is chambers, cerebrospinal fluid is filling that those chambers. So those are a little bit large. And there are neuronal loss, and there are inclusion bodies, et cetera, et cetera. And we can follow those changes by in vivo studies, namely on live patients. We can follow longitudinally using MRI studies, et cetera. The functional and behavioral approach takes neuropsychological testing and the sleep studies and also functional MRI and PET scan, et cetera. And during this meeting, there was a report of progression of adult onset myotonic dystrophy type 1 with a neuropsychological profile worsening. But I think there was a question whether this is overlapping aging effect on top of the static problem that is giving the apparent progression. The molecular, we really don't know how this correlates with the progression of the disease. Are there more nuclear RNA foci as you age? Are there tau abnormality or splicing abnormalities changing with age? These are hard questions you cannot really address in a human being because we don't have those findings until autopsy material become available. But in animal models, those can be addressed. So those are the brain MRI findings, and there are white matter lesions you can see in the area, but particularly temporal pole has been emphasized by French group that may be area of the major involvement. The ventricular sides are generally a little bit bigger, as I mentioned, so the ventricles, and it's a little bit different section, so it's a little bit difficult to correlate, but there are some enlargements. Eyes are very common organ to be involved in myotonic dystrophy. I think majority of the people with myotonic dystrophy develop cataracts, but there was a new interesting findings presented by Mexican investigators, and in that they can spot some streak in the retina, and that can be detected non-invasively. Namely, they don't need to inject anything, but just looking at the tomographic scan of the retina, and then can find this little hyperrefractory spots, and that is present 100% of myotonic dystrophy patients, they said. And so if that is so, this may be very diagnostically important, and also this may be a biomarker if there are some changes with the disease severity. So, but yet this needs to be confirmed with the further studies. The next is disease severity, frequency of symptoms, impact of the symptoms, and the quality of life. Those have to be measured, and we need to identify what matters, what kind of symptoms and disability matters to the patients. So there are a number of efforts done with regards to this, and there is a multisystemic disease severity index being developed by Sensei Gagnon from Canada, and disease burden as well as the health-related quality of life from Rochester Group, and there are some quantitative muscle testing being developed by also Rochester Group and also Canadian Group, and measurement of the muscle changes by MRI of the muscle, and that has been done. And there are more quantitative measures of those clinical outcomes being developed. And in the comparison between the adult onset and juvenile and the congenital onset of myotonic dystrophy, there seems to be the major themes of concern differences between those two groups. In adults, the things listed here, thing listed here are the most frequently occurring major themes, but the impact-wise, fatigue becomes number one, and mobility, etc. And in the congenital and juvenile onset myotonic dystrophy, the profile becomes a little bit different. The communication, education, learning difficulties, slur speech is part of the communication, and hand myotonia, those are the major concerns. So that when we design the clinical trials, those differences matter will matter a great deal. Now, I want to mention about registry a little bit. Dr. Richard Moxley initiated NIH registry at the University of Rochester, and I know many people have registered in his registry, and that's a very valuable tool. Now, since then, there have been also European initiatives. So there are a number of different countries involved, but particularly in France and Quebec, collaborating and making a very formidable registry as well. So in the future, combining all those with a unified worldwide registry, that will be a very important tool for the clinical scientists, and also those will serve as an identification of the people who may be interested in research, clinical research. And it's already going on, and a number of studies have developed based on this population. And one interesting thing is that this is from Treat MND. The research is a major topic of the patients within the registry. So, I think we have a responsibility to answer those questions. Always there's a care and a cure issue. Cure itself is not quite in sight, although we can improve patients' life quite nicely by paying attention to the details. I'm going to come back to that a little bit more details. Now, Dr. Mahadevan already mentioned about biomarkers, so that I'm going to skip that. But it's a nice molecular-based biomarkers are being made. Biomarkers are important because those are sort of an indicator if a treatment is working or is that symptoms are getting better. Truly, sometimes those clinical measures are somewhat susceptible to subjective influences. But the biological index markers are very objective, so that's why it's so important. Now, then we had two presentations, maybe three, with the cardiological complications. As you know, the heart is very important. It doesn't add too much disability in majority of patients with early to middle stage of the disease until late. Then the people may develop congestive heart failure or intractable arrhythmias, and they're going to go into the trouble. But the major threat is sudden death. So here's a heart, and the yellow lines and arrows, so this is a conduction system where the impulse, electrical impulse, is conducted through the heart tissue so that heart can pump the blood in with a very coordinated sequence of contractions of the chambers. The result is on the right side. If you record electrically from the outside of your chest, you can take the electrocardiogram. There's a P wave, QRS complex, and T wave. And the PQ intervals and the QRS intervals are very important indicators for your heart functions. And we can make a decision based on this, and also independently from the EKG, electrocardiogram, the symptoms of recurrent syncope or severe dizziness. We may elect to do further studies. Those are invasive studies called electrophysiological studies, in that they put the electrode through your artery, usually from the groin to your heart, and stimulate the heart and see if those conduction systems are working correctly. And here's if those studies of electrophysiological studies are abnormal, then you can insert the pacemaker and defibrillator. In this case, the middle top is the actual device, and the left X-ray showing that device is implanted under the skin, and then tip is going into the heart. And the right lower area, there's a ventricular tachyarrhythmia, and it's actually the heart is contracting too fast, almost like tremulously, and cannot pump the blood. And usually those people will black out, and if you just do not do anything, they're going to die. So defibrillator kicks in, and then normal QRS complex, normal heartbeat can be restored. If there's a severe conduction block, the similar thing happened with a very, very slow rate of the heart called bradycardia, and that can be treated with a pacemaker. And nowadays there's a pacemaker and defibrillator combination that can help. Now, so the debate happened during this meeting was that whether do we have a really clear-cut evidence that those devices help and save life? It makes sense rationally, but there is no real good hard evidence that those things will work, since those are invasive studies and invasive implantation of the device. Should we take a look at the hard evidence? Now, however, if you do that, we need randomized control study. As you know, randomized control study requires placebo arm. The placebo arm will receive nothing much in terms of the life-saving measure. So this is an ethical concern, and that if you do that, the question has to be truly, we call it equiposed. That means with implantation of those devices and without we really don't know. It's a 50-50 chance of working or not working. Now then there was a retrospective study with Dr. Dubois group from France. They showed there seems to be some improvement in the mortality and lifespan and probability of the longevity with this pacemaker and defibrillator. However, the discussion continues because this study is a retrospective study. That means we are looking back from the chart and extracting the data. And in the process, there would be a lot of biases, and so that may really decreases the authenticity or hardcore evidence of the study. But this is the current recommendations, and you need cardiac, if there is no positive cardiac symptoms, the primary screening of ECG, 48-hour Holter monitoring every second year is recommended, and the echocardiogram over age 40 will be done every 3 years. And this is recently published in International Journal of Cardiology. But if there's a positive symptoms or electrocardiographic abnormalities, then you may go to EP study, that electrophysiological studies. Now, finally, probably one before that, there is a topic about cancer risk. This is raised by two groups. One is a coalition between the Finland and Sweden group, and another one is from National Cancer Institute of NIH from the registry of University of Rochester NIH registry. And both studies indicated about 2 times increase of the cancer risk. And it depending on the organ systems, but it can go up to several-fold higher risk. However, the sample size, in spite it involves over 1,000 patients, but still I think that numerator is very small, and we need confirmation of this cancer risk. But this is a significant findings. I think you need to know about this. And that's here, particularly endocrine and ovarian cancer and brain cancer, colon cancer, those things are on top. And finally, just I was discussing with Dr. Moxley, and this was not really extensively discussed in our scientific meeting, but we really need tissue sampling and repository. European investigators have started doing this several years ago, and the United States, there's no concerted effort to do this. So biopsy tissue, blood tissue, and incidental surgical tissues, and importantly, autopsy. Nobody wants to talk about death, but when your loved one is getting sick, you need to discuss this, and the autopsy material will be really precious, particularly in correlation, to make a correlation with number of different animal models Dr. Mahadevan talked about. So I'm going to stop here. And Valerie, you want to introduce the next speaker?
Thanks very much, T, for that presentation on the clinical developments over the last few days. I'd like to welcome to the stage Andy Berglund, who's going to talk to us about some of the recent developments in turning what we know now about the basic science and more about the clinical course of the disease, to what progress we're making into taking these advances to developing new treatments for the condition, which is obviously the ultimate aim of all of the research that's going on around the world. Andy.
So thanks. This is a real honor. I'm sort of a newbie to the field. Well, I am a newbie to the field. And so I did a little history just to get a feeling for, since I'm giving an overview of the therapeutic sessions, I wanted to see how this had evolved over the years. So what I did was I went back and looked at the previous meetings, and as you can see here, we're definitely increasing the number of abstracts and the type of research that's happening. So I should say I didn't go back to the initial International Myotonic Dystrophy Meeting 1 in Paris, because by the time I hit 4 and 5, I was down to 5 abstracts. And some of those abstracts I might have been pushing a little bit for therapy. But what was really clear today, and there's just to tell you up front, so I may be pulling on other people from the audience, actually, the therapeutic stuff happened this morning and finished about 2.5 hours ago, so I'm not going to give you a ton of details because I didn't quite have time to put it all together. So if you have questions, feel free to ask myself or any of the other scientists here, and we can fill you in on it. So the take-home from this slide, though, is that it's dramatically improving, and that's what I'm trying to show, is that the numbers are going up, and actually the 26 abstracts, depending on how you read certain abstracts, you might even put them, you can increase that number. And actually there's several research groups that aren't here, so I know there's additional research happening. And so it's clear all the basic science that's happened and the clinical research. And these, actually I should say, all these feed in, and this is a great meeting because of that, because we're seeing the basic science, we're seeing the clinical research, and we're seeing the therapeutic research. And of course all these form a nice cycle, and by having us all together and having the patients here, it allows us to, every 2 years, do this cycle and see how things are improving. Okay, so it's definitely getting better. So maybe the next slide, please. Oh, whoops, actually I'm doing this, sorry. Okay, so an overview for the therapeutic design. So I'm sure all of you are aware that CTG or CCTG repeats is the basis of the disease. And then it goes to the RNA, CUG or CCUG repeats. And then that RNA folds up into an RNA hairpin-like structure. And that toxic RNA that's, so the hallmark for this disease, affects muscle-blind by sequestering muscle-blind to the toxic RNA. And then in a mechanism we don't fully understand yet, CUGBP is upregulated. And actually, things we heard and Manny discussed is that other proteins are involved, but these two definitely are at the core of things. And then the change in these proteins affects splicing abnormalities. And then that, and actually, again, what we heard here and maybe in previous meeting, is that it's not just splicing abnormalities. There's other things here, but as it is, it's complicated, so I stuck with just splicing abnormalities here. And then that leads to clinical manifestations. And I should say, Manny Manhaven was kind enough to give me this slide just a few hours ago so I could put this together. And the key here is when you're looking at this slide is to realize that there's lots of places we can target this process, this disease. And as we sort of have this triangle theme going on here, and as you go to the end, there's all sorts of things we can target, and we've heard about that in the clinical highlights and at other times. And there's been some progress on treating some of the clinical manifestations. But really where we want to be is at the very upstream part there. It'd be great to target the CTG or the CCTG repeats and change it at the DNA level even. But that other triangle here, I guess the bottom triangle, the technical ease of intervention, is extremely challenging. So that's why it's such a point up there. But there was some exciting research that we heard from the Pearson lab that is more basic science, so not so much therapeutic design, but giving us insights into potential ways we might be able to target the DNA level. And maybe you could imagine shrinking these repeats. And actually, a few years ago, both Darren Moncton and Christopher Pearson had this idea and screened, but I think it might have been a little early. We didn't have enough knowledge yet about the basic science. So this is an example where by getting a better understanding of the basic science, it informs our therapeutic strategies. So hopefully, by doing the basic science that Christopher and others are doing, will help with that. And maybe the triangle at the end there will get a little bigger. And then again, so the middle triangle there, the potential impact, makes sense again because if you can hit it at the earliest point at the DNA level, right, you'll have a dramatic impact because you're shrinking the repeats. Of course, that's very challenging because you may not want to. I mean, if you're changing DNA, you really just want to change the repeats and not all sorts of other DNA. But the RNA, I should say, is where a lot of people are focusing, and there's some really exciting research that's happening there. So at the RNA level, and I'm sure most of you are aware of this, the antisense, the morpholinos. Oh. Okay, sorry. I'll try and stand a little closer. Okay, so I'll tell you about targeting the RNA primarily, although there's a little bit of work that was discussed here targeting downstream targets as well. Well, I'm going the wrong direction here. Am I supposed to point this somewhere? Oh. This isn't a funny joke. There we go. Okay. Okay, so the categories of molecules that we use for potential therapeutics, I just thought I'd give you a little overview here, and maybe a lot of you already know this, but I just thought it would be nice to have a visual for this. So the three primary types of molecules that are being used at the moment are nucleic acid, and there's lots of different nucleic acids, and sort of the whole range is being tested, and there's some beautiful studies looking at how the modifications affect uptake into the cells, how you can affect localization, stabilization of the molecule, how long they last in the cells. And so that's just a representation actually of the CUG repeats up there in the blue in the corner. So the nucleic acid that we can use and modify, and it's a wonderful strategy because it's exquisitely specific. So you can use the specificity of base pairing to target with great specificity. So that's a real plus for the nucleic acid strategy. There's other strategies out there, the biologics, proteins, peptides. we heard a exciting talk this morning from the Arturo group where they had screened for peptides in a Drosophila model, and they identified a peptide that targets the toxic RNA, appears to release muscleblind, and reverses some of the splicing defects, and reverses the myotonia to a certain degree. So that's an exciting new area, the peptides. And then the proteins, I'll talk a little bit later, and Manny mentioned that as well. And small molecules is something that I think a lot about, and that's, so I forgot to say that. So down in the lower corner is a protein representation, sort of the biologics, a little bigger. And you could imagine overexpressing muscleblind, so that's been discussed. So that's a crystal structure of a part of muscleblind for overexpressing muscleblind and using that as a potential therapeutic. And then something that my group and others think about is small molecules. So that up there, the red molecule is pentamidine. I'm not sure I got everything to scale, but I was trying to do a little bit of scale there. And the idea there is that, and a lot of drugs that are on the market are small molecules, and so we felt like that might be an area that we should explore and others are exploring. But I should say the nucleic acid stuff's a little further along. Okay, so. Okay, I figured out the system. So here's, and you've seen this a little bit already, but I just want to talk about this in a little more depth, so that RNA is where we're targeting a lot, and we use a lot of different technologies, and it's great because there's so much going on in other fields that we can bring into the myotonic dystrophy field for targeting the CUG repeats. So there's ribozymes, so RNAs that will actually recognize and cleave the RNA. So that's been pioneered. Antisense is something that we heard a lot about here, and oligonucleotides, morpholinos, and they're all using nucleic acid to recognize the CUG repeats or the CCUG repeats. And then it's sort of the same thing there, either targeting the CCUG repeats or CUG repeats for the two different types of myotonic dystrophy. And a lot of the readouts we use are seeing how we're affecting the toxic RNA, looking at the foci to see if we can decrease the foci or break up the foci. But you can imagine targeting the proteins as well. And so I mentioned overexpressing the muscleblind protein. Tom Cooper's group and others have shown that you could potentially block the phosphorylation of CUGBP, one of the other important proteins in this pathway, and potentially modulate its levels as a therapeutic strategy. And it's been shown nicely by the Thornton lab and others that you can target the specific splicing defects to correct the missplicing and then reverse the symptoms. For example, myotonia is the one that's been shown very nicely, where you target one specific splicing, you can reverse that splicing defect and rescue the myotonia. And then sort of further downstream back to the triangles, we can target the clinical manifestations, and there's work being done with central nervous system stimulants and mexilatin. Okay. I guess I'm just going to have to be challenged with this. Oh, there we go. Okay. Maybe it's these pretty pictures I put in. So something that I wanted to highlight is, and others have mentioned this, is that it's really important not just to focus on one particular system, but by using multiple systems, we can learn a lot. And so here, and we saw this at the meeting this morning, is that by using Drosophila, by using zebrafish, and by using a whole variety of mouse models, and I'm sorry for those of you that I didn't get all the mouse models up there, these were some of the ones I saw in the talks, is that we can do single molecules that we're really excited about for screening, but for zebrafish and Drosophila, they have the power to do high throughput screening in them. And so we can generate models for myotonic dystrophy, and then we can do the high throughput screening. And so they're really powerful because things are conserved. You can get the toxic RNA in there, the muscleblind proteins are there, the CUGBP protein, and a lot of the other conserved factors. And we can use these to do drug screening. And so it's really important that we, again, support all these models and maybe even develop additional models potentially. Okay. And so, again, something that we saw a lot of at the meeting is using cell-based assays to do high-throughput screening. And just a few of the labs doing this. David Brook did some really nice work looking at a variety of different antisense compounds to see how they were taken up in the cells, to see which were more effective, which were less effective. And so these are just an example of cells that, in this case it's HeLa cells, but lots of different cell models are used to look at how the uptake of compounds, antisense, even biologics can get taken up into the cells. And because a real challenge for us is maybe we can get things to work in vitro with purified systems, then we need to get things working in the cell base, then we go to the animal models. But we want to use all of these again so that we can understand how things work. And so the idea is with these cell-based models that by understanding how things are happening at the fundamental levels of the pathway, we can set up novel screens to identify compounds that if we were just targeting the CUG repeats or just targeting one or two proteins, we might not get. So this allows us to think about targeting additional pathways. For example, one of the pathways is, this was a talk from the Hausman lab, is targeting to splice out the repeats and have the repeats degraded. That's something that's going to be coming online soon. So, and something that is, I've only been to a few of the meetings, but something that's really struck me is the engagement with industry. Is that, and I may have missed a few of the companies as well, but it's quite clear that ISIS and Prosenza and Genzyme have all really gotten involved in this, and it's great because ISIS and Prosenza have this, great technology. They understand how this stuff works, and they're bringing their technology to move these compounds forward. And so there's some really nice collaborative work happening with industry and academics to target the toxic RNA, the CUG repeats, and taking advantage of 30 years of development of antisense for other diseases, but bringing it to, sorry about that, bringing it to myotonic dystrophy. And it's great to see this. So they're bringing their expertise to developing the compounds, collaborating, sharing with people. And again, I think this is important for moving forward to clinical trials. Okay, so I'm sure this is something that lots of families are wondering about, and how soon or how long is probably more realistic until some of these promising approaches reach patients. And just to, so for a successful drug, it takes approximately 15 years to go from research scientist to an approved drug. And that's for the successful drug. So there's all sorts of drugs or compounds people are working on, but you don't know what's going to be the successful drug. And so the challenge is to have lots of compounds being studied and pushing them along, and so that we can get things to move along. But maybe not having too many, but picking the right ones and moving them forward. So I guess I just want to sort of temper our enthusiasm, because what we did see is that with, for example, the antisense, we saw that the antisense technology can target the toxic RNA, completely reverse the splicing defects, reverse the myotonia, and impressively, it lasts for a long time. One of the things that sort of blew me away today was that for a year after the treatment with the antisense technology, that the splicing was still reversed and the myotonia was still rescued. And that was something that I sort of had, before seeing this, wondered about how often you'd have to have antisense treatments and how much you'd need to use. And so seeing these types of results, it's really exciting and encouraging. But again, this is in the mouse, and so we have a lot of work still to do. Okay. But So with the antisense, we are moving forward, and at least this is what I heard this morning, is that it's planned for the nucleic acid antisense, is that it's going to go to, or it's hoped to go to clinical trials in early 2013 at Rochester, at the center there. And so, but I know Prosenza is moving along as well. There's probably others out there that maybe I don't know about, but it's clear that this is going to be something that does happen at some point in the future, I believe. So that I think is really exciting, and hopefully gives you guys some hope. The other thing that's exciting is that by doing this basic science, by understanding all the targets and thinking about things, there's something that's already in clinical trials, a protein antibody that is for targeting a protein ligand interaction tweak that Manny mentioned in the first talk, that we could potentially adapt to myotonic dystrophy. And so that's also exciting. And again, that highlights doing the basic science to inform the types of clinical approaches we can take. And I believe that might be it. So, but one thing I wanted to say, and just two things. One thing I should say is that this meeting 4 years ago actually really motivated me to do more translational research. So my background is sort of pure biochemistry. It didn't even really occur to me to think about doing work that that could maybe have an impact relatively soon. But after coming to IDMC, I guess it must have been 6, and interacting with families, it actually motivated me to go back to my lab and look at compounds that, I thought maybe they could do something. And that actually is what so I guess what I'm trying to tell you is that as families, at least personally for me, you've had a dramatic impact on the way I've done science, and I think in a good way. So I guess what I'm trying to say is you guys should keep interacting with us. And one thing that, maybe this is just me, I'm actually a little intimidated by the families, because I feel like I want to move as fast as I can, and sometimes I feel like we're not going as fast as we can. And so you should realize that maybe you're a little intimidated to talk to us because we're PhDs or MDs, but I think we're just as intimidated. So we should all work on talking about our, the science and how to move things forward. Okay, now whatever it was, the second thing's gone.
Okay, so we're going to get started with the question and answer session, and we'd like to get through as many questions as possible. We know that many of you have already submitted questions on the 3x5 cards earlier today, and we're going to get through as many of those as we can, but we'd also welcome other questions from the floor, and we've got some people with microphones who are out in the audience, and they'll be circulating, so if you've got a question, raise your hand. And we want everyone to use the microphones because the session is being videotaped as well as being streamed, so we want to make certain that the questions are captured, and the same thing for the responses. So those of you who are responding, wait till a microphone is available to you. So what we're going to do, I think I'm going to have my co-chair join me up here, Darren, and we've got a lot of the questions that you put on your cards, and I think we're going to get started with those. And we'll probably go back and forth with questions from the audience and questions from the cards, and we'll ask our experts in the audience to help us respond to a lot of these questions.
So I have the first question here, and it says, I understand anticipation, but ours appears to be both first generation and isolated. Is this extremely rare? And what is it that causes this? So I think I'm understanding the question here, that what we see with anticipation is we see the symptoms become progressively worse from one generation to the next. And most families become diagnosed when that severity reaches a point that the symptoms are severe enough that it leads to some sort of clinical diagnosis. Very often that's when there's a severely affected child is born, or where someone with maybe the juvenile or the adult onset form of the disease, the symptoms become severe enough that they get a diagnosis. And then you go back and you find in the previous generation, oh yeah, there are some symptoms there that kind of get explained by what's happening, and you go sideways and you find maybe there are some other siblings, some uncles, aunts, etc., that may be affected and explain some of the things that are going on. So very often from one individual you end up with several individuals in the family being diagnosed. But then you go back to the next generation, what was happening before that? And maybe you go back 2 or 3 more generations and you can't see anybody obviously with the symptoms. So the symptoms were obviously very mild, or it looks like they may have even been absent. So you might think, well, this just came from nowhere. Where did it start? We're seeing the end point here where everybody's severely affected, but what was before that? So what we now know is that actually it goes probably a long way back. Myotonic dystrophy is pretty much absent in sub-Saharan Africa, but it's relatively widely distributed around the rest of the world. So in all of Europe and Asia, down into North America and South America, the native populations there all have myotonic dystrophy. And what we know is by looking not just at the number of repeats, but the DNA variation around the repeats, we're able to link all of those disease-causing mutations now back to what we call a common founder event. So this is probably someone who probably lived around about 50,000 years ago in a population coming out of Africa. So this mutation is not present at all in Africa. There was probably a change, probably from something like about 5 to 10 repeats up to probably about 20, 25 repeats, something like that. That event probably occurred once in human evolution, with one exception of an African family that T and Ralph Krahe described many years ago. But that probably most individuals are derived from that one founder person who lived 50,000 years ago and has spread his repeats throughout the world subsequent to that. And probably what's happened over the many hundreds, thousands of generations since then is probably from 25 repeats, occasionally it went up to 26, occasionally up to 27, gradually getting bigger and bigger. And it may have been many thousands, tens of thousands of years before any of those repeats became big enough that they suddenly got up to 40, 50 repeats. And then when they get to that size, things happen very quickly, and it goes very quickly from 50, 70, 100, thousands of repeats over a few generations. But the steps before that can happen very slowly. And so what that's telling us is that essentially all of the individuals with myotonic dystrophy in this room probably share a common ancestor. Certainly that going back to that one ancestor 50,000 years ago, but many of the families will go back not that far, but only maybe 4 or 5 generations. You probably won't know about it, and again, others will be connected by maybe 10 or 20 generations. So almost certainly none of the families here are isolated. They all likely go back to a common ancestor in the relatively recent past for human, in terms of human evolution. 50,000 years ago is still relatively recent. So probably nobody is isolated. You all share a common founder. Do you want to do another one? Or? Yeah.
Sure. Maybe I'll ask Dr. Moxley to handle a couple of questions. So maybe we can bring a microphone up here. So we have 2 questions that are very similar. Can individuals with myotonic dystrophy, either type 1 or 2, 1, donate blood, and 2, be an organ donor?
Well, let me ask others to chime in. I don't feel like I'm a world authority on it, but usually with organ donation, you're looking for donation of tissues that have normal function. And as you know, some of the tissues in myotonic dystrophy, although we don't have any major derangements, let's say the liver, for example, there are hepatic enzymes that are elevated in myotonic dystrophy, and we don't really yet know exactly what the significance of that might be. And I would be hesitant to encourage patients to donate a liver. And since we know that there's tissue mosaicism, as Dr. Monckton was saying, and that there are different places in the organ systems that are affected by the repeat, even without a documented abnormality, I would be hesitant to say, yes, go ahead and donate a kidney, even though we don't have clear evidence of dysfunction of a major sort in the kidney. And certainly we already know that, unfortunately, The lens is a major target of pathology. It would be difficult to donate a lens. And other tissues like the lung are often compromised directly or indirectly because of muscle weakness. And we've already heard earlier from Dr. Berglund about the problem with cardiac conduction, even at a subclinical level. So I'm a little nervous about encouraging people to be an organ donor. On the other hand, I would be very enthusiastic about what Dr. Ashizawa was saying at the close of his remarks just a moment ago about the opportunity for patients to help us with donating tissues. And that's a different but a related matter. Now, the first question you asked me, I've sort of forgotten, but it was partly because it was a little trickier for me.
Blood donor.
And I really don't have a strong feeling. Let me just point out a couple of things. Some patients with myotonic dystrophy, either one or two, have a decreased total white blood cell count. Some of them have a mildly decreased platelet count. You can read about this in articles by Chad Heatwall that were published within the past year and then 3 years ago. Some of the patients with myotonic dystrophy have even a lower red cell count. Having said that, other than just these alterations, the biggest other problem is there's a mild decrease in many patients in immunoglobulins. Doesn't necessarily mean that patients with either myotonic dystrophy type 1 or 2 have some important immune problem. It's just that it's a finding. And so I guess on bias, I'm a little nervous about telling patients, sure, go ahead and donate blood, not a problem. And it's because that I'm a little more concerned about the use of the blood and the recipient than I am. I don't think it would harm a myotonic dystrophy patient to donate blood. Now, maybe other people have had the experience that patients have had no trouble, but I would worry about the cells that are involved in the donation and what problems might occur. And it's not simply just that there's a low total white cell count. Many years ago, colleagues in Utah showed that white blood cells have some alterations in their function. You know, they're insulin receptors on certain white blood cells, and probably they are misbehaving a bit.
Okay, I'm going to pick up on, we've just got another question from the floor that relates very much to the previous question. The first question that we had up here. What would be the approximate percentage of the myotonic dystrophy in the population? So the standard figure that we use is around about 1 in 8,000, and that applies pretty well across most of the world, most of Europe and Asia, etc. But as I said before, it's very, very rare in sub-Saharan Africa. And then there are a few populations, such as the Saguenay region in Quebec, where again there's been a founder effect, where that was a population that was established probably 300 or 400 years ago with a relatively small number of individuals, and one of the individuals in that population probably carried a myotonic dystrophy mutation, and because the population was established with a small number of people, the incidence there is much higher, around about 1 in 400. So that's the answer to that. And then I'm going to come back to another question here that relates to the point that Dick just talked about in terms of, as T raised, about how we know some of the things that we know about myotonic dystrophy. So one of the questions is here is how do we measure the number of repeats in the brain and the heart and the muscle, etc. So that obviously depends on the tissue. So the blood is easy. We can take blood samples from many patients. We're also fortunate that many patients offer us muscle biopsy samples, and we can use those to measure the number of repeats. Clearly, however, other tissues are much more difficult to get. So we can't take brain biopsies from patients who are still alive. That just isn't possible. We can't do that. Willing as some people might be, we wouldn't be able to do that. So the only way we know that the repeats are much larger in the brain is from brain. That kind of partly explains why we know less about that, because obviously those samples are just much, much more difficult to brain. That kind of partly explains why we know less about that, because obviously those samples are just much, much more difficult to get. And we want to understand that process. So as T and alluded to there, if there are people who think that they might be able to help us with that, then they should get in touch.
And I think we had a question over here.
Not really a question, just kind of a point of interest in terms of donating blood. I used to do it on a regular basis. There wasn't any problem for me, but they finally told me that they were going to stop taking my blood because of an increased liver enzyme that was present. So my count for that liver enzyme was always like 5 points high. And I went to the doctor to check out and make sure that there was no exposure to hepatitis or things like that. And he said, no, it's just because of the myotonic dystrophy that enzyme runs high. So they didn't take my blood for that reason. So just a point of interest.
Thank you. Yeah. We have a
Just a quick.
microphone, please.
Oh, just a quick question about the 1 in 8,000. Is that diagnosed or a notion of undiagnosed, including in that?
Yeah, that's a very good point. So that's kind of what we think in terms of individuals are diagnosed. And clearly that's an underestimate for the true number of individuals. So if you think about it, as I said, most of the families that we now know about have someone who's relatively severely affected. And maybe it's someone who is 30 years of age and has just been diagnosed. That's the index case in a family. 5 years previously, we knew nothing about that family. That family was still there, though, and many of the individuals in that family will have been having symptoms, and there will have been unexplained things going on. So there are undoubtedly other families out there who are on the edge of being diagnosed, and then there are families following on behind that. So you imagine there are people out there now who are maybe 20 years old, who are not going to get the symptoms till they're 60 or 70, and they don't have any children yet, but their children will get them. So there's this conveyor belt of new families coming along. So at the moment, there's obviously no impetus for us to go out there and do a population screen. But maybe in the future, when we have therapies where we can intervene, and very likely any therapy will be done better before symptoms start, then there may be a real case for going out and screening the whole population to find the individuals who don't yet have the symptoms and start treatment before the symptoms even start. That would be the ideal. Okay, Manny, did you want to say something? You have to get a microphone.
Yeah.
Manny, yeah.
Might just want to, we had a study presented at this meeting today, this week, about congenital myotonic dystrophy, and that was a population-based study, and so in Canada. And from that study, I think the numbers for the incidence of congenital DM were about 1 in 48,000. So that, might give us some degree of accuracy about what the incidence of CDM is anyways.
Okay, I think we have a question or comment from here.
I just had a question. I know that they're doing the trials with mexilotine. I just wondered what it actually does for the body.
What mexilotine does for the body?
Like what symptoms it affects and what.
Do we have somebody who's involved in that trial? Dick? The effects of mexilotine. Here, share this one.
As most of you know, mexilotine is a derivative of lidocaine, and it acts on tissues to influence sodium current, but indirectly it influences the movement of chloride ions. And over the years, it's been found to be very effective in treating myotonia, delayed relaxation. And so since there are more data that support the idea that in both DM1 and DM2, the myotonia is due to an alteration in the production of the proper amount and form of the chloride channel, if you will, both of these are chloride channel myotonias. And there's been a lot of evidence clinically, anecdotally over the years that patients with chloride channel myotonia got better with mexilotine. And so recently, our group published a paper in the Green Journal of Neurology showing that mexilotine was very helpful in a controlled, randomized, double-blind crossover study of mexilotine given in a dose of 150 milligrams 3 times a day, and then in a follow-on study of 200 milligrams 3 times a day. And so we've found that if you take mexilotine with food, you block most of the significant side effects. We did not encounter any significant other side effects. We monitored patients very carefully during this time, and now we're embarking on a randomized trial of 6 months duration. The earlier trial
But many of these young adults who are having some major challenges in employment and all kinds of other ways, I would venture to say their challenges did not begin last week or last month. That they have been struggling with a lot of these symptoms since childhood. So in light of trying to hasten the diagnosis of juvenile onset, is it accurate to say that CNS involvement, including cognitive and neuropsychiatric problems, is the primary symptom of juvenile onset and not neuromuscular symptoms? I mean, what do we need to do? There are lots of studies out there now. Do we need to extract and interpret what is already out there to agree or disagree with this? In true adult onset, many are spared the buckshot-like assault on their brains and lead very productive lives until adult onset symptoms appear, like, of course, like the Christmas tree cataracts. Now, this is my older son's EKG the day he was admitted to the emergency room, and his heart rate, he will probably correct me on this, it was 230 beats per minute, and then within about 48 hours, he had an implanted ICD. And he also takes Sotalol, which I believe has really helped keep his heart regulated. And here we have a very good example of ptosis, or the, one area of muscle weakness. So these adults, later may have some cognitive involvement, including apathy, fatigue, memory loss, among other symptoms, but they were spared the significant CNS involvement that some of our children are experiencing. And then there's just one other point that I would like to make. We often hear myotonic dystrophy described as the most common form of adult onset muscular dystrophy. Well, this makes no reference to the congenital and the childhood forms, and it leaves one thinking that it is a muscle disease, when in fact it is so much more than that. And I do know that medical students spend very little time learning about the neuromuscular diseases. So if the little short definition of myotonic dystrophy is this that is flying around, it makes it particularly difficult for clinicians to really have a grasp of the disease that some of their patients are really dealing with. So I'm just wondering how else we might phrase this, how we can flesh it out a little bit, so at least for the clinicians, that they have some greater sense of what our disease is really like. The pediatric neurologist who finally diagnosed my older son after 7 years of searching said to me about 2 years ago, he said, it was not muscle weakness that brought you to my office, it was learning problems, social inattention. It had nothing to do, he said it's very confusing. So I think in our education, if we can make sure we use the best wording to best convey our our condition, it would be very helpful and help doctors make a faster diagnosis. So I would like to, well, you've already seen my acknowledgments, and I want to say especially thank you to my husband Larry and to my son Hunter, who helped me with my PowerPoint. I'm still like one who has difficulty climbing up the steps and chewing gum at the same time. This does not come easily to me. And in addition, I'd like to extend to all of you investigators my deep, deep appreciation for your untiring dedication. And then for all of my fellow family members, may we do everything we can to educate ourselves, and kudos to you for being here, because you are increasing your education, and we need to advocate for our families and assist the investigators as we get closer to these, to treatment trials, and offer biopsies or whatever they need to help. And as everybody keeps saying, this is a team effort. So thank you very much for the opportunity to be here today, and thank you.
Thanks, Shannon. This is why I knew to say almost nothing and to get out of your way. So the next speaker is John Brecka, who's going to talk about adult onset DM.
As Dr. Porter said, my name is John Brecka, and I have DM too. I'm a bit of a Luddite in that I don't have a PowerPoint presentation, so I'm just going to try and talk to you about adult onset. Although I am not the ideal person to talk to you about adult onset, in that I've been incredibly fortunate in my life that I had a career prior to being affected by my disability and eventually retiring, so that, as I said, I have been very fortunate as to the extent of my disability. Before I start, though, I'd like to thank Dr. Richard Moxley and Shannon Lord, because when I first discovered I had DM, being a type A personality, I ran around like a frantic lunatic trying to find out everything I could about the disease. And through their encouragement, I went to IDMC-8, which brought me into the fold of becoming active in the in both the IDMC and the Myotonic Dystrophy community. So I'd like to thank them both from the bottom of my heart, because you guys are why I'm here. As Lisa talked earlier, the adult onset diagnosis is an odyssey for most people. I was fortunate in that my mother had myotonic muscular dystrophy type 2, but was never formally diagnosed, although toward the end of her life she did get a diagnosis that was some form of muscular dystrophy similar in appearance to Steinhardt's disease, which, as we all know, now is called PROM or DM2. She went through the odyssey that was probably 11 years or more in the making, going to various specialists. When she first started going in her early 50s to doctors, well, Betty, you're just getting older. You know, why are you here? Yeah, you're a little fatigued, and maybe your muscles are getting weaker, but you know, it's aging. And since the apple didn't fall from the tree, she had a bit of a type A personality herself and said, that doesn't sound right, and kept trying to pursue options. She went to various specialists in New York. She ultimately was diagnosed at the Mayo Clinic, but in the meantime, she was diagnosed as having hypothyroidism, possibly fibromyalgia, and I'm sure this is going to sound similar to a lot of the adult DM patients here, in that DM, both forms, are frequently misdiagnosed. And that's one of the challenges that I have for those members of the research community. Not necessarily the doctors here, because I'm kind of preaching to the choir by talking to you guys, but the information needs to trickle down to other doctors. I mean, you guys are all mainly at large teaching hospitals, and your staff and your people know what to diagnose. But what happens a lot of times is that information needs to get out to the broader community. As I said, it took about 11 years for my mother to get a diagnosis. after she got her diagnosis, one of the treatments that was proposed at that time was high-dose steroids. And they said, well, since the muscles are getting weaker, give steroids, should help. I mean, it was very good logic, but thankfully she was not treated with steroids because we did a lot of research on steroids and their effects, and she started saying, well, the cure sounds as bad as the disease. I'm getting a little weaker, but there's a lot of other side effects from steroids, but so she didn't wind up going through that, although she did go through a lot of other things. And that's why one of the things that we need to get out to the community at large are standards of diagnosis and more education as to how to properly diagnose patients with both forms of adult onset muscular dystrophy. Because most patients come into their doctors with complaints of fatigue, tiredness. You may have symptoms similar to irritable bowel syndrome. But those things are non-specific. And the medical community does need to be educated more through the efforts of everyone here. And we need to partner as patients to take it upon ourselves to also help educate our doctors and educate those people in our family that may not have been diagnosed yet, and encourage them to get whatever help they need. I was diagnosed fairly easily because of what she had. When I went into my doctor with complaints of tiredness, fatigue, I was having problems rising from a standing position. And we kind of knew what direction to look in. It wasn't, well, you've got all these things. My mother had previously, as I said, been diagnosed with some form of muscular dystrophy. So I asked my father about this, and he said, oh, no, I've talked to the doctors, and it's not genetic, it's just something she has. You get that, I mean, and that's what he said. He said, talk to the doctors, and you don't have anything to worry about. It's in your head. Well, I can tell you Dr. Day has done a brain study of my head, and there is nothing in my head. But after he did that, I went to Cleveland Clinic. And the Cleveland Clinic doctor that I went to was good in that he said, yes, there is a possible genetic cause, and let's start testing for this. I had the unique experience of having probably a 45-minute EMG, because when they did the EMG, they found classic dive bomber noises and said, well, do you mind if we bring in some residents to hear this? Being a fool, I said yes. So I probably had the longest EMG in Cleveland Clinic history. I also had a similar experience when I went to my eye doctor, who's an optoneurologist, that had some residents following him around that day and said, oh boy, subcapular iridescent cataracts. We don't get them too often either. But that was not nearly as painful. I just sat there with my chin in a microscope. But one of the things that they did after they did the EMG was they said, well, let's take a biopsy. There's a gene test for it too, but let's do a biopsy. So they did an open incision biopsy and said, it looks like myotonic dystrophy. You should probably get a the gene test. That took probably about 4.5 months, only because my insurance carrier said, well, they're outside the plan. And I said, well, they're the only lab in the United States that does it. Luckily or unluckily, depending on your opinion, I was a lawyer in a prior life, and I wrote some nasty letters to my insurance company, which resulted in them deciding that it would probably be much more economical to cover the blood test than it would be to fight. I do enough pro bono work for the foundation, Manny. But as I said, I went there with fatigue and tired symptoms. I got a diagnosis of DM2, and that led to my, well, what can I do? My doctor there did not have the world's greatest bedside manner. He basically, they said, well, you got to come in for the gene test results. I said, okay, can you tell me what I have? Well, come in for the gene test results. Okay, so I go in there and bring my wife along. And he said, well, we got results back. You've got myotonic muscular dystrophy type 2. And I said, okay, what does that mean? And he said to me, and I'm trying to quote as accurate as I can, but it's probably a little off after a number of years, but he said, well, it's an incurable muscle wasting condition that's going to get worse throughout your lifetime. There is no known treatment, no known cure. We have no therapies for it. And maybe if you want, you can talk to people at the MDA clinic down at the University of Miami, but I have nothing more that I can tell you. So pretty much, see you, wouldn't want to be you. And I then embarked upon my odyssey, which is how I met Dick and Shannon and a number of you here who I can't acknowledge by name, but I'm incredibly grateful for all of your help and support. I eventually retired from the practice of law mainly because of cognitive effects. I had problems, going up and down stairs and fatigue, but I could deal with that. The problem that I was having is an executive function deficit. It's pretty similar to what I observed in my mother, and it frankly scares the hell out of me. She wound up with what was diagnosed in the nursing home as dementia, but who knows? Unable to recognize her family or pretty much anyone else. And I had noticed that before she was even diagnosed, she had problems with word finding, and I've talked to a number of you here that have also recounted to me similar stories in that it's not that you don't know the words you want to use, you misuse them. You say yes instead of no, or you use other words, but it's like the brain when you're forming sentences goes through and pulls the wrong word out of the rack. And a lot of times my wife will correct me because I don't even realize that I'm doing it. I used to practice appellate law, and appellate law depends upon writing written briefs and oral argument. Written briefs I didn't have a problem with because I can write and go back and correct, and through that process get rid of where I screwed up basically. But I can't go into an appellate argument or into a hearing in a court case and say, well, my client is, completely contests all of these charges, and instead screw up and say, well, my client doesn't contest these charges. It kind of would get me in trouble with the bar. So that eventually forced my retirement. As I said, I've been fortunate in that I had disability insurance through my firm. My income was reduced dramatically, but I can't complain. I mean, I can still eat and do all of the things that I'm used to doing. But I would challenge the researchers that what happens with myotonic muscular dystrophy, fatigue, pain, gastro symptoms, those tend to bring adult patients in the door. But what ultimately becomes as much or more of a concern to them are the brain symptoms, the CNS function symptoms, which I was very glad to hear that there have been a lot of presentations at IDMC on all of those effects. I think I saw the Rochester study, and the only, not really criticism, but comment I have on that is that I think that I'm the ultimate example of selection bias. I can come here and speak to you and sort of coherently relay what my symptoms are and what's going on in my life. Many patients don't have that luxury, and you have to rely on caregivers for that. But I think part of the problem with the survey and using the registry is that it selected people like me that were able to fill out the questionnaire, which is why I think CNS symptoms may have been underreported. But again, that's more for future surveys, because I thought there was a lot of good information in that study. So I'd like to challenge you all here to remember to concentrate on brain function, because I've said this to a lot of you, that if you found a cure for myotonic muscular dystrophy that fixed my brain function, but didn't really take care of my body and I had to drive around in a wheelchair, I could live with that much more easily than if you fixed my body, I turned it into Arnold Schwarzenegger, but had a brain full of jello. I would much rather, now obviously I'd like both. I'm greedy. you know, I'd like to get a body like Arnold and, you know, improve my IQ. But no comments from the peanut gallery. But in closing, what I'd like to say is, like I said, the adult onset is very difficult to diagnose, and we need better standards of diagnosis. We need more in the way of standards of care, because some things that have been talked about previously, such as gastro symptoms, can be worked on. Muscle pain is something that needs to be addressed, especially in DM2, and I tell people I've got it every day. I go into the doctor's office and they say, "Are you in pain?" And I say, "So what's new?" I mean, those symptoms, though, are manageable to some extent. They're not all completely manageable, but they're manageable to some extent. But I'd like to challenge everyone here to remember that drugs that cross the blood-brain barrier are the key to at least hopefully arresting the progression of the disease, if not finding a cure. And together, through the partnership of the patient community and all of you researchers, I'm confident, and much more confident than I've been in the past, that we can find a cure for this disease, or at least a treatment, and I'll settle for that. And I'd like to thank you all very much, because I have been fortunate to get to know a lot of you, and without the efforts of the research community here, we wouldn't be nearly as far along as we are, so thank you.
Thanks. The next speaker is Diane Bade, who's going to talk about the caretaker perspective.
Well, now, as a really good caregiver, I recognize fatigue. You guys kind of wiggle it out a little bit. You've been here for a while. I don't have slides, and I'm going to be pretty quick, okay? Coming from a caregiver perspective, I kind of had to think about that a little bit, in that what perspective are you looking for? Are you looking for me coming from caregiver as mom? Are you looking at me as coming from the caregiver as a wife? Are you looking at me as coming from a caregiver as a professional registered nurse? Those are all entities that are included in caregiving. It's definitely a family disease that we have. It's also a multidisciplinary disease. There is not going to be any changes in diagnosis, in treatment, or in cures without some multidisciplinary collaboration from all people who are caregivers. Whether you're a provider, whether you're an investigator, whether you're a patient or a family member, all of those things must come together in a working relationship to come to answers for myotonic dystrophy. In healthcare, we call it the continuum of care. When a patient comes in the hospital, we look at them and we say, you know, this is their diagnosis, so they go to that floor, and we're going to take care of them. Then they get up to that floor, and we find out, gosh, you know what? They also have those and those things, but we can't take that body in 6 different directions, so we have to take care of the whole person. That's really hard to do, especially when the patient comes in and says, well, it's my gallbladder that's bothering me. That's it, it's just my gallbladder. However, I have mental deficits. I can't get up and go to the bathroom by myself. I need an attendant. There's a lot of other things that go in. Myotonic dystrophy, in my family, in my caregiving experience, I have found it comes with a lot of denial. If you were to ask my husband, who passed away in 2005, had myotonic dystrophy, when I would ask him, how are you feeling? You're not looking real good. And he'd say, I'm fine. And oh, okay, you're fine. So I promised I would write on his headstone, here lies Chris Bade, died of terminal fineness. Because he lived a life that was a balance that was right for him. He didn't see when his oxygen was being deprived. He didn't notice that when we went to the top of of the mountains that suddenly he wasn't functioning as well as when we were down in the valleys, because the oxygenation that was going on in his system wasn't working as well. Well, the caregiver came up with a little pulse oximeter, so I knew when he was not doing good. This disease is one that doesn't follow the script for most families. One of our biggest challenges is that most of our families find out about this disease and about their diagnosis through a crisis moment, whether it's the birth of a baby Kayla. For me, it was a husband that was going in for sleep apnea surgery that went very, very wrong because we didn't know about the anesthetic complications at that time. To my son, who really was doing his best to try to be that football player, but his coach says he's just not got any muscles. I don't know what's wrong with him. We were fortunate enough to, when my son, that coach, brought us that news that says, you know, he's just not growing like he should. Has he ever had a physical? We went to my husband's family doctor. My husband had numerous medical complications, none of them diagnosed as myotonic dystrophy. For over 18 years, we went through many, many things. With the kids, we were at neurologists, we were at pediatric ophthalmologists, and we did exercises for the brain. They jumped on the trampolines and they followed those circles. Nothing ever said myotonic dystrophy. We went into my husband's primary care physician, and he says, you know, this just looks like more than what I know about. This looks different. With all the problems you've had, speaking to my husband, I'd really like you to take Scott to another doctor. I went to school under him. He's a neurologist out at USC, and I'd like you to talk to him. And I'm thinking, neurology, okay, we're gonna go jump through some more trampolines for the kid, because obviously it's part of the learning disabilities that we're going. Well, we walked into Dr. Said Badoon's office, and Dr. Badoon looked at my husband, looked at my son, shook hands with Scott, and said, Scott, I want you to go sit in that room for a minute because I have to go talk to your parents for just a second. And he took us into the other room, and he said, well, I have to do some testing, but I'm 99% sure that your son has myotonic muscular dystrophy. And he looked at my husband, he says, as do you, Mr. Bade. And literally, all of our hopes and dreams just fell right on the floor at that moment. I'm a caregiver. I'm a professional. How could I not know that there's a disease in my family? The devastation that comes with that. And of course, my only association with muscular dystrophy was Duchenne's. And I thought, you're telling me my kid's, he's 15. How could he possibly be something like that? We asked about support groups. Sorry, even MDA. Well, we have support groups. They're general support groups. You go to one of the general support groups and you see these darling children in wheelchairs. And your kid has got a little problem, but he's not in a wheelchair. And you think, okay, that's not me. We're not in the right spot. We go somewhere else. The itinerary for disease diagnosis and process that goes on just falls apart at that point. And being the person I am, I'm one of those that, call it A-type personality if you will, but I'm also one that likes denial a lot. Just don't look at it and it'll go away. I like to bury my head in the sand, as Shanna would say, but my problem is when I bury my head in the sand, I keep getting bit on the butt by all this stuff, you know? And I have to pop back out and say, what was that? What do you mean? The collaboration that goes with healthcare is one of those things that just keeps getting me in the backside. We can get the buckshot that goes through the children's cognitive abilities. We need to get that buckshot out to the rest of our healthcare community. We need to scatter. It's not just a disease of the muscles. It's not just a disease of any one of our systems. It is multi-system. It is multidisciplinary. How do we as family members, as caregivers, as professionals motivate our patients who suffer greatly from apathy, fatigue? How do we get them to be advocates in their own health care? Well, I can tell you one thing. I went to the IDMC in Quebec in 2005, and there were a handful of family members there, and we got pretty excited about what was happening with research. But the biggest challenge was they were saying, how come those researchers are so worried about my hands? My hands don't bother me at all. It's my life that's getting in the way. And at the same time, the researchers were talking to one another and saying, you know, we're finding this, we're finding that. And as you've heard throughout the weekend, and especially today, all of those things are coming together because there is more collaboration and cooperation. We need the long-term health studies. We need people to be engaged and involved. And as professionals from the medical community, we need to encourage those with myotonic dystrophy to stay actively involved in their own disease process. I know it's tiring. I know it takes a long time. But if not for you, for someone else, there will be a difference. Thank you. Hearing the disease treatment papers and that this weekend, you know, there was just no options back then, and I think the 20 years that we have morphed, I look at it, this room, it blew me away to see this many people, affected and non-affected people coming together for one disease entity. And I applaud all of you because it takes a lot of stamina to just show up and do it. And the researchers, I know some of you guys are young fellows and you know, this is what you got to do to get your paper published and get this stuff done. But I hope, I pray, that at some point during this weekend, something triggers that little piece in your brain that says, I want to know more. I want to figure this out. There's something that we've missed, because there is something we've missed. We've known about the disease for 100 years. In the last 20 years, we have made phenomenal growth because of your bright, curious minds. Keep the collaboration going, keep the energy up, and I guarantee everyone will benefit. Thank you.
All right, I'm gonna give you about a two-liner introduction to our last two speakers. They, like the majority of us in this room, are members of families with myotonic dystrophy. However, these advocates are coming from a slightly different perspective than most of us in the room, and that they are coming from the medical and scientific community. Our first speaker is going to be a young woman, Sarah Jordan, who is a first-year medical student at the Cleveland Clinic Lerner College of Medicine. I believe she told me that she knows of 15 affected relatives, including a congenital brother, an older brother with adult onset. And she found me on the web, and we have been communicating and talking to T and Mari, and she has some fire inside of her, and we need that kind of drive and energy. The second speaker, I'll just go ahead right now, is probably not a stranger to any of you now, is Eric Wang, who has presented beautifully at this conference. And like Sarah and like many of us, he also is coming from a family with myotonic dystrophy. And so I think we need to listen to these young people and we're bringing the next generation on. Yes. So Sarah will speak first.
Hi, my name is Sarah Jordan, and this is Eric Wang. And as a medical student at the Cleveland Clinic, I would first like to apologize for any procedures that you have had to sit through. I wasn't sitting in on them. But I also want to extend a huge thank you to the organizers of the conference and everyone like Shannon and Lisa and Dr. Porter who've really helped me along the way in getting here and getting this message out to all of you. So today we're going to be talking about how to bridge the gap between these groups. Okay. So we've heard from a lot of the speakers today in the advocacy section about the incredible struggles that the families are currently facing in reaching diagnoses. Ridiculous examples of decades that people spend searching, and Dr. Moxley's frankly disturbing numbers of over 6 years, and the difficulties faced in the multisystemic nature of the disease, and how this isn't, you don't go straight to a neurologist when you're having gastrointestinal issues or ocular issues. And so we really need to increase awareness in the general population, but especially in the medical community specifically. And in addition to that, we've also heard from so many amazing research groups this week who are doing some really exciting work. But we need more. I worked for several years in labs in muscular dystrophy, and there was a prevalent belief that I noticed that the funding for Myotonic was somehow limited, that it was easier to get research funding if you were looking into diseases like Duchenne's. Now, this is completely incorrect. It's understandable to see how they might have thought that if you just look at the numbers. For example, in the fiscal year 2010, the research in Duchenne muscular dystrophy received approximately $38 million from the NIH, as opposed to myotonic muscular dystrophy, which only received approximately $12 million. However, these numbers are limited entirely by the number of investigators that are submitting research applications and grant applications. So what we really need is an increased number and a greater variety of researchers in the field. So how can we do that? Investigators, talk your colleagues into DM research through collaboration. It really is the best way to bring people in. Another really important role you play is as a mentor to young investigators, from undergraduates through postdocs. The trick is to get them while they're young, but then, really help them develop into successful DM researchers themselves. And in the interest of making collaboration and bringing fresh minds into the field as easy as possible, we are working with the Myotonic Dystrophy Foundation to create a database with information that researchers often need, especially when coming into a new field, such as funding sources, cell and animal models, and review papers on all of the relevant topics. So we sincerely hope that if you have something to contribute, that you will send it in to myotoniceducates@gmail.com, and we can get started working on that. And based on the brilliant suggestion by a researcher here this week, we are also looking to create an investigator directory. You can think of it as a DM Facebook. Basically, you would just send us your areas of interest, the institution you're currently working at, and your contact information. And then when you have some interesting eye findings, but you're a muscle pathologist, you can click ophthalmology and find a list of people to collaborate with and work with, and we won't have any really significant findings falling through the cracks. So that was investigators, now advocacy groups. We need to increase visibility in DM specifically within the general population and the medical and research communities to really get these individuals interested and wanting to get involved. It's really just getting the name out there. You also play a crucial role in providing seed money for these young investigators so that they can go on and one day run entire labs that are focused on DM research. And patients, get involved in your local advocacy groups. Get active in fundraising. Ona McConnell is a brilliant example of what one person can do. Last year, she raised over $50,000 for DM research. It was enough to fund a postdoctoral fellow for an entire year of research. Yeah, I don't know, is she here? And there's one thing that everyone in this room can do to, one, increase the number and variety of researchers in the field, and also increase awareness amongst physicians across all specialties, which, as we've heard, is a huge issue. And that is education. So at my medical school and many others, we have what are called patient experiences, where an investigator comes in and they give an introduction to a disease and its background, and they also talk about the research in the field and what they're currently working on. And then a patient comes in, and sometimes with their families, and they give their perspective on their own experience with the disease. It really allows the students to learn about the disease in a way that they never could from a textbook. And it sticks. Our very own Dr. T. Ashizawa first became interested in myotonic dystrophy research when a patient came to speak at his medical school. And Shannon Lord and Dr. Hopkins are already currently doing an annual program at Emory School of Medicine. Our goal is to see programs like this at medical schools all across the country. I'm currently submitting a governing council action item to the American Medical Association, so we are going to have that entire network of medical schools to work through. But if we want this to become a reality, we need your help. We need investigators, physicians, and patients to go to www.mden.weebly.com and register. Let us know you're interested, and then we'll do all the rest. We'll coordinate with your local medical schools and arrange the program. It's a very small commitment on your part. The talks are just an hour or so. And oh, and we're giving patients a template of topics, so you won't have to worry about figuring out what to talk about. And soon we'll have some more resources available for you on the website, such as example templates for presentations and a video of an example of a session. So we have a small, it doesn't look small, there's so many people in this room, which is wonderful, but we have a relatively small but very tight community here. So we're really counting on a really good turnout from the people gathered in this room today in order to really get this program going. So in conclusion, if we are going to combat this awful, awful disease, we need to get physicians and investigators more aware and interested. And this educational network is really the way to do it. So I hope you'll go online and register today. And if you have any questions or concerns, comments, or even ideas, you can shoot off an email to Myotonic Educates that I mentioned earlier, or just grab me in the hall afterwards. I'd love to talk to you. Next, Eric is going to talk about his personal experiences as coming from a family with myotonic, and in his words, why he urges you to support efforts like this.
So thanks, Sarah. My name is Eric Wang, and it's an incredible honor for me to be here as an advocate for myotonic dystrophy. Earlier in the week, I gave a talk as a researcher who studies myotonic dystrophy. Today, I'm speaking to you not only as a researcher, but as a person who grew up seeing the effects of DM every day and watching how it has impacted his affected and unaffected family members. When I look back on the past 15 years of my life, there was a clear turning point in my relationship to DM, how it affected my family, and how I dealt with this fact. Throughout junior high school and high school, I watched my dad gradually lose fine motor control, lose the ability to get up and down stairs, and go through several close calls with his heart and severe falls. In college, I remember how every time I came home for holiday breaks, I would notice how my dad looked a little bit different each time as a result of the muscle wasting. The disease impacted me and my family in a very negative way, and in general, we felt very helpless as to what we could do to slow down progression of the disease. After college, I worked in a lab where I studied cardiovascular biology. During that time, I applied to graduate school and had to choose the area of research where I wanted to do my PhD. I consulted many other students in the lab about interesting research areas, and there was one particular evening that really served as a eureka moment for me. I was having dinner with another student in the lab, and I remember it very vividly. We were in the basement of John Harvard's pub in Cambridge, Massachusetts, and I remember that we both ordered a mushroom Swiss burger. And it was a delicious burger, and not because of how it tasted. It was because I remember that that night was when a light bulb went on in my head. It was there that my friend suggested that I study DM to, quote, be a champion of the disease. In a way, the idea was extremely obvious to use my biology training to study a disease that runs in my family. But for some reason, I had never considered it seriously. I think it was partly due to a narrow way of thinking that I couldn't possibly make a difference. To me, this narrow way of thinking is something that I'd like all of us to transcend in some way. That night, my journey of empowerment began. I later found David Hausman at MIT, who, along with many other members in this audience, found the gene for DM1 in 1992, and also sought out Chris Burge, an expert in computational biology and the study of gene regulation, whose recent entry into the DM field has already led to advances in our understanding of DM. I also sought out many other members of the DM field who have served as informal mentors to me over the past 5 years. And let me tell you, the DM field is absolutely remarkable for its collegiality, its drive, and its focus. And as a result, throughout my graduate career, with the help of all these individuals, I've been fortunate enough to try to make contributions to this field and hopefully make an impact. So to investigators,
if you take mexiletine with
I'd like to give you a
food,
message that
block
we need to
most
bring
of the
other
significant side
investigators to the
effects.
field
We
with
did not
complementary
encounter any
expertise that we will need
significant,
to move things
other
forward.
side effects.
This means
we monitored
talking to your
patients
colleagues who study
very
other
carefully
aspects of
during
biology,
this time, and
our
now
industry
we're
partners,
embarking
and
on
those
a
in regulatory
randomized
bodies.
trial of
To
6
conquer this
months
disease, we will
duration.
need,
The
as
earlier
many have
trial
described over the past few days, to work together as a team and together with patients. DM families, many people with or without DM go through life not finding something that really drives them, something that they live or die for. Obviously, DM in my family has been a curse because of its terrible effects, but on the other hand, DM has really given me an incredible purpose in life, a challenge and an obstacle to overcome through which we will all do our best to prevail and become better people. I urge you to take a moment to imagine a world in which you or your family still has DM, but you aren't limited by any practical or financial concerns. What would that world look like? Is it a world in which all doctors know about DM? Is it a world in which people on the street understand what a DM patient looks like and what their needs are? Is it a world in which teachers at school know how to deal with the needs of a DM child? Is it a world in which genetic testing for DM is free in the US? I'd like us to all think about what we can do, each one of you, to get closer to that world. I'd like all of you to try and believe that you can, because if you don't even think you can, it's not going to happen. This, I think, is the adjustment in thinking that we all need to make, to dream big, and of course, to later come back to reality, but to have that courage to dream. Progress in the DM field has been phenomenal, but we have not yet reached the end of the road. There will be many hills and valleys and numerous storms to weather. We need to be prepared for these moments, but we must also be prepared for that inevitable day when therapies are a tangible reality. We must organize so that we can move forward carefully, yet deliberately, with a passion tempered with prudence, and we need to support and encourage one another so that we can get through all of these experiences in one piece, and we must make it our goal to continue pushing on in spite of all odds. We will overcome this disease, but it will need to be a group effort. In the way that DM has served as a paradigm for diseases of RNA toxicity, let's show the world our paradigm for how to work together to cure a terrible disease. Thank you.
Wow. What a day. My name is Roy Swanson. I'm from the University of Florida, and with T. Shizawa, we organized IDMC-8. And I guess I'm absolutely amazed about this community right here. Just absolutely incredible. So it's my job as the very last session to do something very pleasant. This is the time of year when I try to convince my kids that it's better to give than to receive. And usually I'm met with great skeptical looks, but we all know that's true. And following on what Sarah and Eric just talked about, this is the next generation of scientists who we plan to celebrate in the next few minutes. So I'll ask you to hold the applause until we get through all of the recipients. The first set of recipients are people who have presented posters who were judged by our scientific community as being outstanding. And for each session, we have 2 poster winners. So I'll read those off, and actually, I guess it's better than that at Christmas because I get to unwrap the presents and give them. So our first poster winner is for our session on disease mechanisms, and it's poster number 4 by Isabelle Bem-Asmont from Christine Brandlatz and Nicolas Sargent's lab. Isabelle, are you here? This is fun. Okay. The second award winner is also for disease mechanisms, and this is Fabio Martinelli from the University of Milano. Fabio? Fabio has my toast. The next award is for cell and animal models, and this is being presented to Dr. Ron Batra from the University of Florida. Actually, just Ron Batra. I don't want him to get, he's actually my student, and he's still a PhD student, so I don't want to be premature. Ron? This is for the, this was actually a very difficult section because it was clinical, it was a biomarker section, it was also covered, as Shannon will be happy to know, just the brain. And so this is awarded to Luc Hubert from Jacques Quimerat's lab in Quebec. Luc? So this is being presented to Jack. Where's your student? Here, Jack. Jack. The next one is in the same section, clinical biomarkers and the brain, and this is Jim Hilbert from the University of Rochester. Jim. All right, and now the last section of the meeting, which was therapeutic development. We have Maud Christine Chouinard from Jean Mathieu's lab in Quebec. And then last but not least is Aaron Foff from Mani Mahadevan's lab.
You have to go home and take care of the kids.
Okay, and then now I get to turn it over to Lisa Harvey, who will be presenting the Myotonic Dystrophy Foundation Award for Excellence in Research Awards.
So for the last several IDMC conferences, we have gone, and we wanted to honor the scientists who did the best presentations. The criteria for these awards is to either be a postdoc, a graduate student, or a researcher new in the field that's less than 3 years in the field. And so, our hope is to do that, by doing this, that we can help to encourage people to join the field and hopefully stay in the field. So, our first winner is Catherine Hagerman from the University of Rochester. And her presentation at the IDMC conference was Gene Expression Biomarkers of Disease Severity in Myotonic Dystrophy Type 1 Muscle. I wish I understood that. So these are each $1,000 awards, and then the plaque will be coming in the mail. The next award goes to, I'd love for you to come up together, and that is Eric Wang and Thomas Wang. Eric Wang's presentation was a transcriptome regulation by MBNLs via splicing and non-splicing mechanisms. Wow. And Thomas is splicing of the CUG repeats in the DMPK 3' UTR attenuates DM1 pathology. I'm so glad I'm not in charge of research. I have have to say, the interesting thing about the awards that we gave, we had a scientific peer review committee select these from all of the people in that group. And it's interesting that it turns out both are families with myotonic dystrophy, which I find to be very, it's interesting that we're all here together. And so I just very excited to see this group coming together, and I see a very bright future for everybody. So thank you.
So next award is from MDA, MMD Support Group. And this plaque says, it was your lone myotonic dystrophy voice that spoke volumes to researchers. It was your fearless desire to put the face to MMD that helped bring MMD education and research to where it is today. Thank you from the MMD community of researchers and families for your continued support and education. And this person is very special person who made this IDMC meetings bring to today's status. And also, she was instrumental in bringing the patients to the scientists. And this is Shannon Lord. Please come up to. So I tell you a little story. After IDMC-2, she came to Baylor College of Medicine and presented a check of $180,000 approximately, I can't remember the exact number, to support research or research-related activities. So we decided to support IDMC using majority of the fund. And to date, it's been very useful to keep this series of meetings going. And she joined us in IDMC-3 in Kyoto. But prior to that, there was an American Society of Human Genetics meeting where she showed up with Larry and 2 children. And they were introduced to scientists. And this was the first time that DM scientists mingled with the patients. And that was the trigger of this interaction between patients and scientists we can see today. So congratulations, and thank you very much for your contribution.
So I've been kindly invited by the Myotonic Dystrophy Foundation to present the award for lifetime achievement in myotonic dystrophy. And it's a real honor for me to be able to even present this award. And I have the award here, if I can get it out without dropping it. And I'm just going to read what it says here. So this award is for meritorious service to the myotonic dystrophy community. And the recipient of this award is Professor Sir Peter Harper. Many of you will... I know Peter won't thank me for doing this, but I'm going to make him wait here a few more minutes and just say a few more things. So quite clearly, nearly everybody in the room is already fully aware of the momentous contribution that Peter has made to myotonic dystrophy research. but I'm gonna draw it out a little bit longer and just remind you a few of those. So many of the patients and families will be absolutely familiar with Peter's book, the Facts About Myotonic Dystrophy, which set out in plain and simple language what the consequences of the disease are and how we can best go about dealing with the condition. And that's obviously the culmination of Peter's many years of experience in actually meeting and helping patients and families to manage their lives. Obviously, absolutely vital information to help patients and families. Peter obviously has also made a tremendous contribution to our basic understanding of this particular disorder. Peter started his research on myotonic dystrophy in the early 1970s. He did working out here with Victor McCusack, as he told us earlier on this morning, going around and visiting many patients and families in their houses. He used that knowledge of meeting families to go back to the UK, where he was a human geneticist and clinical geneticist at the University of Cardiff, where again he continued to see many families with myotonic dystrophy. He made major contributions in understanding the congenital form of the disease and establishing that the role of the affected mother in transmitting the condition. Lots about the early efforts to map the gene. So Peter was involved in establishing that the mutation was indeed located on human chromosome 19 and linked to blood markers. And then throughout the 1980s, Peter made major contributions. His group was at the forefront of the mapping efforts, narrowing down on chromosome 19 till eventually the gene was cloned in 1992. Throughout that time, he's been instrumental in trying to ensure that the benefits of understanding the genetics of the disease are actually taken back to the patients and families in terms of the use even before the gene was cloned, in terms of linkage analysis, how that might be used in a predictive manner, and obviously subsequently, since the identification of the mutation, how that can be used in genetic counseling to help families and patients make informed choices about how their lives and reproductive choices. And since then, obviously, Peter has continued to work on myotonic dystrophy, and the culmination of much of his efforts over the years are encapsulated in his book, his monograph, Myotonic Dystrophy, which is for many of us the Bible of understanding this particular condition. It's been immensely useful to new researchers coming into the field, graduate students, clinicians, etc. All use this book as a major resource for understanding the disease. And a point here is that the last edition of the book is now getting a few years old, and Peter a few years ago asked T. Ashizawa, myself, and Charles Thornton if we would take on the honor of trying to update the book. Unfortunately, that several years have passed since then, and you'll notice there is not a new version of the book. And it's T and I and Charles have come to realize what a monumental task that Peter has undertaken to produce this monograph. And we will endeavor to take things forward in the future, but we will likely be calling on many of you in the audience to help us with that. So it's really brought home to us what a tremendous amount of work has gone into producing that. Obviously, we're talking today about myotonic dystrophy. Many of you may not know, but Peter has a parallel life where he had as significant a contribution towards Huntington's disease, and again has produced the book on Huntington's disease, as well as obviously over the years being involved in genetic counseling and ensuring that the new knowledge deriving from human genetics is brought as quickly as possible to the benefits of patients and families. He was for many years on the Human Genetics Commission and Advisory Committee on Genetic Testing in the UK. He was awarded a knighthood for his services to human genetics in 2004, and since he retired in 2004, he continues to work both on looking at the disease, but also an increasing role in ensuring that the research history underlining the recent developments that have gone on in human genetics during Peter's lifetime are preserved. Peter has been practicing at a time when we knew very little about human genetics, and there wasn't really much we could do about it for many of these conditions through to getting the diagnosis, and now the exciting stage where there are actually new treatments coming online. So Peter has really been instrumental in that process in both myotonic dystrophy and in Huntington's disease. And I think there's no doubt that when we're we're talking about a Lifetime Achievement Award. Peter has devoted 40 years of his life to researching this condition and is an absolutely 100% deserving recipient of this award. So it gives me, again, great pleasure to congratulate Peter and to present this award to him on behalf of the Myotonic Dystrophy Foundation.
All those surprise things, you know. On behalf of the Myotonic Dystrophy Support Network, a specific support group of the MDA for myotonic dystrophy, it's my pleasure on behalf of all of us, families around the world, to thank this particular individual for the foresight, bringing together world researchers, working together for a common goal to find treatments and cures for myotonic muscular dystrophy. Dr. Ashizawa. You know, having the support group and the researchers coming together every year for the last, I believe it's been 13 years, maybe thereabouts, Southern California Myotonic Dystrophy Support Group has been privileged to have researchers and colleagues that have come to us as family members, as disease recipients, if you will, what a privilege, come together and share their knowledge as they do with you now. But the fact that we, through Dr. Ashizawa, now have the privilege of coming together in a forum like this, where families, research, and community of myotonic dystrophy comes together, it's just phenomenal. Thank you so much. I just look forward to 2 years from now when we're going to find out what poster really gets produced. Thank you so much.
Thank you, Diane. We only have a couple, I actually only have a couple more presents to unwrap. Actually, I have one to unwrap and T has one to unwrap. So when we started organizing the meeting, we looked back at all the comments that people had made about previous meetings, and there were a couple of comments that for the first time in Würzburg for IDMC-7, we had awarded something called the Hans Steinert Award to celebrate somebody who contributed particularly strongly to both basic science and clinical science. So it's my duty, my proud duty, to present the award for basic science. And when T and I got together, we tried to decide how we were going to decide who to pick. And then, of course, being the strong chairman that we were, we decided to default to our scientific subcommittees and let them choose. And then all we had to do was add up the votes. So that's what happened. On the basic science subcommittee, of 8 individuals voted for people they thought had contributed the most in the last 2 years to the myotonic dystrophy field in basic science. And today's recipient in basic science is someone that I've respected a great deal for a number of years, who's contributed to so many different facets of myotonic dystrophy research. He's a clinician scientist, so I respect clinician scientists enormously. They manage to do clinical work and on the side manage to do science. And the science this individual has done has been truly outstanding, truly remarkable. He has bridged the gap all the way from very basic biochemistry to deriving one of the most popular animal models to coming up with some really spectacular therapeutic approaches that we heard about today. So it's my honor to present the 2011 IDMC-8 award to Dr. Charles Thornton.
So next award, the equally important Hans Steiner Award for outstanding contributions to myotonic dystrophy research and care for the clinical contributions, and that goes to Dr. Jack Primera. All right, I think this concludes our award ceremony, but I want to have a little final remark. Now, if you can put the slide for the potential next venue. With your approval, I think we're going to go to IDMC-9, and the sole candidate site is San Sebastian, Spain. This is a Basque country, beautiful, beautiful site, and very historic. And Dr. Juan Botas, who is, I think, he's half Spaniard, half American, but he said whenever the soccer game comes on, he's 100% Spaniard. But he told me, amazing thing that we can't really deliver it to you by the picture is that city has a truly amazing food. So anyway, so I would like to just open the bid. This is only the site, but if you, in the audience, if you have any volunteering site, just please stand up and name it. And I don't see anybody else. So unfortunately, the organizers, the future organizers, there are two of them. One is Dr. Anthony Udisberia, and another one is Dr. Adolfo Lopez de Mnain. And Dr. Lopez de Mnain is a long-known myotonic dystrophy researcher in Spain, and both are Basque people. And as you know, recently the Basque went into truce with Spain, and the region is 100% safe. That's another important consideration factor. But so then I think we can say IDMC-9 venue is the officially the San Sebastian in Spain in 2013. Okay, great. And so finally, I would like to acknowledge our special thanks for wonderful work to do the legwork and all the important heavy lifting for this meeting of the IDMC-9 part. That's the University of Florida team led by Kathy Gentleman. Please raise your hand. The team, entire team. Okay. And one final comment. I wish Margaret Bowler could have been here, and she, unfortunately, her health didn't allow to come to this meeting, but as you know, Margaret is a leader of a UK myotonic dystrophy support group, and my heart was with her, and I'm quite sure she watched the streaming, and she knows what's going on. So Dr. Harper, please convey our message that we missed her very much, but the next meeting I would like to have her. It's going to be very close from England to Spain. Well, so this will conclude the IDMC-9 meeting, and thank you very much for your participation and support, and really I'm glad to see the advancement we made. But thank you.
It's been a long day. You all have been very patient. Now it's time to party. Is anyone ready for a drink besides me? So
we need to talk to
hold off on that drink.
Is Mr. Bach here? Yeah, he wants to say a few things, so just wait a second, and then I'll give you the podium. I'm sorry, I forgot to mention about this.
I wanted to speak from a patient and family perspective because I was really impressed at my first IDMC how many really, really smart people I saw working on myotonic dystrophy. And I'm not sure that you hear it often enough, but I wanted to give the patient and family community a chance to say thank you for working on myotonic dystrophy.
Well, that was certainly well worth another minute. This has been a historic day. Never before in history have this many researchers, patients, and families come together with one common purpose. We come back in an hour here. We'll be transforming this historic day into an historic luau. 7 o'clock. If anyone has any questions that have not been answered, please fill out the forms. We'll have another chance tomorrow. And please, please, please take this survey, fill it out, and drop it off before you leave tomorrow. Thank you. See you in an hour.